So I'm going to be talking about a test called ELISA, which stands for Enzyme-Linked Immunosorbent Assay, and it's a test done with a sample from, in this case, a patient's spinal cord fluid, and it determines if a certain pathogen is present in the patient. So here's how the test is performed. So you have these wells, they're really small wells, and a sample is added to them. And in the sample, there's proteins, there's a bunch of cells, just a bunch of random debris. And in the well, the sample, if there's a pathogen, it will bind to the walls of the well because it has antigens on the surface, and the antigens stick to the walls of the cell. Other proteins will also stick to the walls of the cell. And after the sample is put in and it sits there for long enough, then there's a wash, and anything that's not stuck to the walls is washed away. So next, the researcher will add a primary antibody, and this antibody will only attach to the pathogen that they're testing for. So it will not attach to other particles. In this case, the pink particles are the pathogen, so they would attach to that. Next, a secondary antibody is added to the sample, and the secondary antibody will, first there's a wash in between that step. So any of the extra primary antibodies will be washed away. And then the secondary antibodies are added to this sample, and then they link up with the primary antibodies. If there's no primary antibodies, they wouldn't link up with anything. So these secondary antibodies have an enzyme attached to them, and you'll find out why in this next step. So in between the two steps, there's another wash and anything unneeded, anything that's not attached to the pathogen, anything that's not the secondary primary antibody will be washed away. So in the next step, there is a substrate added, and the substrate is specific to the enzyme that's attached to the secondary antibody, and it's going to attach to the secondary antibody, and it will turn the well a color, in this case, dark blue. So the dark blue indicates that there's a presence of the pathogen in the sample, and if there was a lower concentration of the pathogen in the sample, then it would be light blue. So if there was less pathogen present in the sample, if there's lower concentration, then it would be a lighter blue. So that is how they detect for the presence of the pathogen in the sample using ELISA. If there wasn't any pathogen in the sample, I'm going to show you what that would look like. So if it was just some proteins in the sample and the primary antibody was added, it's only looking for the certain type of pathogen that it's the antibody for, because these primary antibodies are extremely specific. And if it can't find the pathogen, when the wash step occurs, they're just going to be washed away. And then when the secondary antibody comes in, they're not going to find the primary antibody, so they're just going to get washed away too. And then when the substrate comes in, they're not going to find any enzyme that's linked to the secondary antibody, so they'll get washed away too. And that would make the well clear, which would be a negative result.